The protein prolyl isomerase. What in the world is the protein prolyl isomerase? What's the big picture about that one particular protein? What are we talking about? What's the big concept? It's not necessarily an enzyme. It's a catalyst. Okay, it's not. It doesn't fulfill a reaction. It doesn't do an organic reaction. So it's not necessarily an enzyme. Okay. What was the bigger picture we're talking about in this section? Sometimes we get in the details and we forget where we are in the big section of this chapter. It's about proline and it's about structure. It's about proteins, generalized proteins, being able to fold in its correct structure. Okay? And we said ribonuclease, under experimental conditions, can do it all by itself. Just the interactions of the amino acid side chains can make sure that the fold comes up into its active form. But later we found, as you get larger, there are places where aggregates can form or misfolding can occur. And they are called kinetic traps. They fold, and this little part of the molecule is really, really stable. But the rest of the molecule isn't as stable as it can be. And so we have to get past that fold either by giving it energy or by using a helper, okay? A helper to catalyze movement. We looked at the first helper was the, the one that shuffled the sulfurs, okay? And so the PDI, the protein disulfide isomerase. And so if by chance a disulfide bond forms and it's the wrong one, that covalent bond has to be broken and reshuffled, and we had an enzyme to help that. The second thing that we're looking at is proline, because proline has two conformations, one trans, one cis. This is a better way to look at the cis. And you can see the polypeptide, the chain of the protein goes, gets to the proline, and then it comes right back, and it puts the two chains right next to each other. The trans would push one of those chains way up, and there's less steric hindrance. How often do we see cis in proteins? A lot, a little. A little. Okay, about 6% of them are in cis conformation, and they usually are there to provide a structural component that we need for protein function. And so sometimes they're in trans, sometimes they need to be in cis. So this protein goes to the other proteins that are trying to fold, and it will bind to prolines, and it will allow rotation to occur. Now, why in the world does the bond that is that arrow, why is that not rotatable? That's a single bond, isn't it? The nitrogen to the carbon is formally, we write it as a single bond, right? And then the carbon to that red oxygen, we write it formally as a double bond, a carbonyl. But remember what happens in a peptide bond? We have what? Resonance. And so the blue nitrogen to the black carbon usually has partial double bond character because the electrons are resonating. What this protein goes and does is this. It will bind at the proline. It will interact with those electrons and stop it from resonating. Okay? And if it stops it from resonating, then the nitrogen to carbon does behave like a single bond, can rotate, and so it facilitates that rotation. Now, it'll only go into the cis conformation if all of the rest of the protein gives us more stability than the instability we get by having those two chains being close together. So it's a combination between all of the rest, and we know the few amino acids that are right next to that proline are going to be negatively. You're going to have steric issues, right? But if the rest of the protein is okay, that can overcome it. Okay? And so this doesn't necessarily change all trans to cis. It just allows rotation to move. And if everything works out better, it can move it. Okay, binds to that amide, the carbon, oxygen, nitrogen, and it can allow the rotation. All right. Now, if you think about proline, Prolyl isomerase, it will help proteins fold. Okay. It also can be a regulator of proteins. If you think about it, three-dimensional structure of proteins dictates what they do. All right. If I have a protein that has like this, trans, that's a little bit different than if I take this entire chain and push it down. That makes a big difference in structure. And so this can also be a regulator of protein function because I changed the structure so dramatically. All right. More complex helpers in the folding process are under a larger category called chaperones. Chaperones are larger proteins that bind reversibly to segments of proteins that are in the midst of folding. Okay, they use ATP, and what they do is they break apart aggregations. Okay, now you're going to have a little bit of aggregation as the protein initially is synthesized, because the first thing that happens in proteins is that hydrophobic collapse. The hydrophobic goes in and tries to get in the middle, right? Well, if you're in the middle of making your protein, you don't even have the full chain out. You've got lots of hydrophobics that are just floating around, and they tend to aggregate. And if you don't have the whole sequence, parts of them can aggregate in ways that you don't necessarily want. They have to be in different things once you get the full chain out. So sometimes we glob up together because I'm in the midst of folding, okay, or I haven't even synthesized the whole chain. These proteins will go in, use ATP, the power of ATP, just to open things up. And so what will happen is it'll go open it up, and then the protein will try to fold again. Sometimes it'll fold in a different way. And if it folds in a good way, then if it comes and opens it up, it'll fold right back. But if it's in a bad way, if I open it up, it moves towards the better. And so it keeps opening pieces of that protein up until it can get to the final conformational fold. Again, thermodynamics is driven by the interactions of the protein. It wants to get to the most thermodynamically stable interactions throughout the entire protein. All right. Now, here is a slide that has DNA K and DNA J. These are proteins identified in E. coli. They are homologs of proteins found in the mammalian system called HSP70 and HSP40. The HSP are called heat shock proteins. The 70 and the 40 are indicative of the size of the protein. Okay? And so these proteins were found when people heated up cells. These proteins upregulate. Okay? Well, what happens if you heat up a cell? What happens to the proteins? Do proteins like heat? No, they start to unfold. They start to denature. These proteins are here to help the cell recover and make sure the proteins get folded back. Now, we call them heat shock proteins because that's where we first found them in that response, but that's not their normal response. The normal physiological response is when synthesis occurs, these proteins are working, okay? But they get upregulated, higher regulated, or we get more of them under heat conditions, and so that's when we first found out what they did. And so when they first got what they did, they were named, and now we got lots of proteins that are named one way and actually are used totally differently. And so, but the heat shock proteins, that's what happens. So what happens is an unfolded protein will come and bind to either heat shock proteins or the DNA KJ system, and they bind together. The J and the K, or the 70-40, create a workable unit. They bind ATP. They break ATP. They cause conformational movement. The protein gets moved. They refold. The proteins dissociate. The heat shock proteins dissociate. I may get a partially folded protein. It may be fully folded. If it's fully folded, it goes away. If not, those can bind back in different places. Again, use ATP to open it up just a little bit. allow it to refold, it dissociates, and it goes into this cycle until, if I unfold it, it folds right back to the same place, and it's fully folded. And the power of ATP, I'm using chemical energy to cause physical movement to allow the good fold to get to. Another class, and I've swapped the order of slides, so don't worry about this. This is an animation. I will show you the animation. Much larger class, the chaperones, and multiple subunits. We don't quite understand the mechanism because it's so large. The proteins are so large, but this is what we understand about it. We understand there's no sound. Okay, so a protein got placed inside of the top of this very large protein. There's multiple subunits around, creating a barrel or a hole for proteins to get down into it. Then a cap set of proteins will bind to the top of it. Okay? Along with that, 7 ATPs are bound. Now, as the top takes in a protein that's unfolded and caps it, did you see what happened at the bottom? 7 ATPs were let go and another protein was let go at the bottom. So it coordinates top and bottom. As things are bound in one is released, and so it works back and forth, moving back and forth. And once it's in there, those ATPs are used to unfold it and allow it to refold again. And that's the process. Here is the pictures of what happens. And so you can see the unfolded protein gets in there. On this side, there is a much more folded protein. The cap comes in and binds. There are 7 ATPs being bound. When this conformation occurs, it moves this protein such that it releases the cap. The protein that was being folded falls off. The ATP is released. That allows this function to actually occur, and it hops back and forth. The protein that needs to be folded can bind and rebind multiple times until it reaches its fully folded process. If you look at the last slide, the H-shot proteins can do some of the folding and then send it off to this mechanism working together. So sometimes some of the folds need the heat-shot proteins, but some of the other parts of the fold might need this chaperone to get the complete fold. Now, these are large proteins that need help. Okay, so you're folding very large proteins. That means these folding chaperones are huge to accommodate a very large protein inside that cavity. All right. And again, up to about 15% of proteins, even in E. coli, need this mechanism. And E. coli is a relatively simple genome with proteins that aren't that big, not nearly as big as mammalian systems are. Okay. Let me do this. Oops. I'm going to change this slide just a little bit so you can see. All right. I'm going to do this. I'm going to do this. Boom. Okay. I just deleted stuff. Okay. I'm going to make this bigger. This now bigger, not smaller. Oh, that's smaller. Oh, that's bigger. Okay. Here's the question. Now, I said proteins fold until they reach the most thermodynamically stable state. All right? But proteins are not absolutely thermodynamically stable. Okay? It might be the lowest energy state, the best energy state, the best interactions, but proteins are not at their absolute rock bottom thermodynamically stable conformation. Why in the world are proteins not at the most stable it could ever be?
If they're completely stable, they wouldn't react with it.
That's exactly it. Biological processes, even though we show pictures, are always dynamic. Okay? That means if a protein is fully stable, it's a rock. It wouldn't interact with anything else. But in biological processes, what has to happen is you have to have some flexibility because you have to interact with other things. You have to open up a little, create a binding pocket, interact with what's going on, to bind something, to allow a reaction to happen, to unfold and let it go. If it doesn't have a little bit of instability to allow it to interact, we would lose all functionality. And so, even though proteins will fold to what we call the most thermodynamically stable conformation, there's still a little bit of instability to allow us to move. Okay? And what we have to do is be at a very good balance point for the temperatures of our body to still allow that protein to function and to be movable. All right? And so we have to have this. And even in its lifetime, it has to be at least opening up enough so that it can be degraded and recycled, because we have this process of aging and recycling of our protein. So there is dynamic ability to do things, which really is not great to look at in a textbook because we don't have a lot of things that are very dynamic in a textbook. We kind of get this static picture of what's going on, especially when we look at crystal structures. It's a structure of a protein that is in crystalline form like a salt. We don't have proteins in our body that look like salt. Okay? But we can take good pictures of them in an instrument if they are crystallized. Much better than some of the techniques we have in physiological conditions under what we actually have functioning in our bodies. All right, we have come to the end of the slides and the information. What I do want to do is go ahead and talk about the article and the quiz. All right, here's the actual quiz. All right, what I'm going to do is extract 1, 2, 3 of the figures in the journal article. Now, here's the deal. In journal articles, they are going to provide you the experimental conditions and the results to convince you of basically the story they're laying out. This is the data. This is what it means. Okay? And so reading a journal article, what you have to do is you have to understand their premise. What are they trying to say is happening? And then you have to understand the experimental conditions and look at the data. And does the data say what they are saying it says? Okay. And so they're leading you down a path. Are you going to go down that path completely? Or are you going to look at the data yourself and say, well, what about this? That doesn't quite make sense. Or absolutely, it makes sense. I was reading an article just recently about education, and here it said, in K through 12 system, 82% of students cannot distinguish between an advertisement and a news article. Okay? Talking about being led down the path. They do not have the ability to distinguish a claim, an advertisement claim, and a general news article that has been vetted and has some validity behind it. Now, the article also went to say, don't just blame the kids. 59% of adults fall into the same category. And so, being able to critically think and evaluate what is being said is important. Just because it's posted on the internet does not mean it has any validity. Just because it is written well doesn't mean that it has validity. Okay? Just because it's in a peer-reviewed journal article doesn't mean, although it's supposed to have been vetted, maybe has a little bit more credibility behind it, but you should be skeptical and look at the data yourself and see, does the data actually support what the author is saying? Sometimes it does. A lot of times it does. And sometimes there are things they don't talk about that they probably should have, which might put into question the conclusions that they come. So I'm going to ask you about 3 different figures. Now, reading a journal article is a wonderful process. Okay. Sometimes when we read journal articles, it might take, you and every third word looking up what that means, and, sometimes they're tough, right? They're not easy reads. Now, they will give you overviews, but they're giving an overview to what they believe is an audience that studies what they study all the time. Their focus is very narrow about what is general information. They assume you know a lot of things, all right? And so, when you get to a journal article, this segment right here, what is this called? The abstract. What is the purpose of the abstract? Brief overview. Conclusions, this is what we found. Okay? And that's their claims. In this, there is no data to support their view at this moment in time, but it's supposed to give you an overview. Then followed by what is the next section? The introduction and the background. This is where they give you the background of the general area in which they're supposed to be looking at. Okay? Quite often it gets really, really technical. It's not as general as you would like. I actually have, at some points I have an assignment where I have you compare what the textbook would cover and what a general background would cover in a journal article. They talk about the same thing. They sure don't look the same. You can understand what the textbook says, but the background seems to be way too technical. Okay, so this is helpful when you read, okay, the background. The next section is, materials, experimental methods. Sometimes reading the experimental methods is like reading the book of numbers. Okay, it is very technical, and quite often, when you do the genealogies, you just kind of zone out. When you get into, we use this protocol from this person, unless you actually use those protocols very often, even what they reference, you've got to go to the reference to actually figure out what's really going on. Sometimes the methods and materials aren't as important to you when you're just trying to get information. If you actually have to repeat the experiment, that's really important. But that is probably less important to you extracting good information than some of the other pieces of the puzzle. Okay? You might need to look back and read a little bit of it, but that's not the primary source that you start looking at. It's got lots of figures. Okay? You get to the next part, and depending upon how the journal article is organized, you get results section and a discussion. Sometimes you get results and discussion mixed. Okay? The results are supposed to be an explanation of what you see in the data, and then the discussion is the context. Sometimes they combine them, sometimes they separate them. And so if you're looking at 3 figures and you see this results section that goes on for several pages, do you have to read every bit of the several pages? No, what you want to do is focus in on the areas of the article that refer to the figures in that article. So by doing the quiz, I didn't say review the entire article, review the portions of the article that I feel you should be able to get some information from. So this article is much longer than what I want you to engage in. I don't care that you understand all the parts about the NMR and the sequencing data and all of that. That's not important to what I want you to get from this. Okay, so this has a results section and a discussion. So for figure 2, you may see some information in the results and information in the discussion that is important to explain what's going on. All right. Okay. What I do want you to look at is, okay, in generalities. Okay, I asked you to do figure 2. Here is figure 1. All right, let me make this, let's see if I can make it bigger. The experiment here is kind of odd, okay? And so you see a squiggly line and 2 humps. Okay, what does that mean? All right, let's look at the axis. Temperature, okay, got that. and excess heat capacity. There's not even a number on that axis. Okay, there is a scale for in kcals. All right. So we're going to take a protein and we're going to obviously heat it, right? And so in this case, this experiment had it over a range of pHs. What we are seeing is there is an absorption of energy in two distinct places. Okay? There's a hump where I absorb energy, and then there's a hump where I absorb a lot of energy. Okay? What the information that you derive from this experiment is this, that when you have an absorption of that particular heat or that particular energy, what's happening is you're getting a conformational change, a change in the shape of the protein. Okay, so there is two very distinct transitions under these experimental conditions. Okay, that's the experiment and what they're deriving their information from. Okay. All right. This experiment. Does anybody know what this looks kind of like that we saw in lecture? A what? Titration? It's not the titration curve. It is the experiment with circular dichroism, which measures what? Okay, I have failed utterly as a professor. You don't remember circular dichroism? Let's see. Okay. Circular dichroism uses polarized light to shine through a sample, a liquid sample, and the polarized light gets twisted. And what's twisting the polarized light in a protein structure? The chirality of what particular secondary structure? Alpha helix. And so an alpha helix has a dip that is characteristic with 1-2 dips in this range. Now, they're going to show you this part of the graph, and they talk about beta sheet. I think that's a little sketchy myself. Alpha helix is the better measurement, okay? And so what you're seeing is 3 distinct conditions, 1, 2, and 3, and you can see what's going on in those conditions in the experimental. And they're going to show how much alpha helix content is found in each one of those 3 conditions. All right, what you would expect fully unfolded with no alpha helix should kind of have a straight kind of line up through here, maybe just a little bit of a dip. The larger the dip, the more alpha helix. So that's the context of what we're looking at there. Okay.
What's that called again?
It is circular dichroism, and they call it CD, just to confuse everybody. It's the CD spectra of. They assume you know exactly what that is. Okay. In this experiment, there's fluorescent spectra of 50 micromolar ANS. Okay, so ANS. Oh my goodness. This is where you go back to the experimental conditions here, the methods and materials, and this is actually you get some information about it. ANS is a organic compound. Okay, I think they actually define it somewhere, but it's an organic compound that does this. It is fluorescent if and only if the organic molecule is surrounded inside a hydrophobic pocket. So it has to be surrounded by a hydrophobic pocket. If it's just in water, it doesn't fluoresce. Okay. If it is associated straight up to a hydrophobic surface, it really doesn't fluoresce much. It has to be embedded and surrounded by a hydrophobic environment before it can fluoresce. That means absorb a wavelength and then emit a wavelength. And so they're going to put it in a fluorimeter that can excite at 380. So they use 380 nanometer UV light, and then they recorded this spectrum between 400 and 640 nanometer to see what comes up. And so if you actually look at there's the 400 to 640, we see the maximal absorption is somewhere around 480. So we excite it with 380, and really we take the 480 as the measurement of how much is being absorbed. And so there are, in the last experiment we had 3 states. In this experiment we have 3 states of the protein plus a control. And then the control here is the spectrum of ANS with no protein. And so it's just ANS and water. We would expect ANS that has no hydrophobic pocket to be in should have no absorbance, and that's what we see. So that's control. That's why we have 4. All right? And so, based upon these 3 different pieces of data, along with others, the argument of this group is that they can, let's see, what does it say? They show the structure and thermodynamics of extraordinarily stable molten globule state of canine milk lysosome. All right? And so they isolate the protein lysozyme from milk, from dogs. They said, this particular protein is the best example of an extraordinary state of molten globule so that we can study it. Much better than anybody else in the entire universe. And basically, if you read them, they kind of have that kind of attitude. Oh, here it is, ANS. 1-anilo-naphthalene 8-sulfonate. There it is. That's why we use ANS. And so, I would like you to look at the data set, what the data is. I don't care what the axis is. I know what the axis is. I want you to explain why that data supports their statements. Why each one, why the dips were the way they were, why the look of the peaks in one of them, or why the absorbance was different between each state. That supports the idea that under their experimental conditions, the right pH, the right temperature, they get a good molten globule that is better than anybody else's. Okay? So that's what I would like you to do. All right. Go do that. Have a good day.